Hi, this is Alexander from CovidGraph, and in this video we will show you how you can find information to the ACE2 receptor, the angiotensin converting enzyme 2. What you see here in this screenshot is the receptor the coronavirus uses to enter the cell. And we are interested as researchers to find as much information to know more about the virus and how to prevent the infection. Usually what researchers do is either go to Ensembl database to find ACE2 as the gene. We will do that here. And then you select here the human entry and you can find information for the name. Here you see the Uniprot identifier where you can see the coding protein from Uniprot. You see that there are 2 transcripts here from RefSeq, and you will find additional information. The other source for gene is the NCBI gene browser. I prepared that here already, and you have the angiotensin 2 converting enzyme as an entry. And when we click on that, you will get also here the information. Here you see the Ensembl identifier, and so on and so forth. Usually what you do is you take the gene identifier, go to Uniprot, and enter here your gene of interest and do a search. I did that already, and what you see here are 13 already reviewed entries for coding proteins of ACE2, and there are several more, 470, that are not reviewed right now. Sometimes you're interested where this gene of interest is expressed, in which kind of tissue. There's one database from the Broad Institute called GtexPortal, and you can go on the website here and also look for ACE2 the gene, and you see here the specific gene expression in the specific tissue. What we do now is the same exercise of browsing for ACE2, but doing that in our CovidGraph. And we start by typing in look for gene ACE2. And here you already see that you can use semi-natural language queries to find gene entries. And here we have ACE2, which is, according to this color here, is a gene symbol, and this corresponds to the Ensembl gene ID. We can do a right mouse click and find the synonyms of the gene, which is here ACEH. We can find as well the mappings, which is the first jump from one database to the other database, which is here between Ensembl and NCBI gene. So we have that here, the NCBI gene. We can do the mapping as well, the forward and the backward mappings. And here we see already that this is kind of a complex structure. We have mappings between the different databases, between the different names, and we try to get information to that. Now, when we are interested in the transcripts, we go here to the gene ACE2, we do right mouse click, and we say what is coding. And here we have 5 transcripts from NCBI, so 5 different transcripts from one gene. We can take them here as one and do again the same. We ask the question, what proteins are coding? And here we see that these 5 transcripts result in 8 different protein entries in Uniprot. And what we've basically done here is we jump between NCBI, RefSeq, and Uniprot and make that available to you in one place and have that available and interconnected to each other. When you're interested now in a specific tissue of expression, you can use another query here that we prepared for you, which is look for tissue, let's say lung. And When we look for that, we get here the lung tissue, and we can use that here now and ask the question, is there an expression between ACE2 and lung? We are doing that by selecting those two nodes, do the right mouse click, and say here, shortest path, which will calculate the shortest path between the two nodes in this network. And this is a very easy example here because these two nodes are directly connected by one relationship here called expressed. When you're interested in other tissues where ACE2 is expressed, you can also go on this gene here, do the right mouse click, say expand, and follow the same relationship type to a tissue. And we are doing that now. And you see that ACE2 is expressed in a lot of different tissues that you can all see here in yellow, which means that the coronavirus basically attacks much more than lung or heart that we knew so far. And with this, I hope I could show you how easy it is to browse different data sources within CovidGraph, to have everything in one place and to have easy queries and an easy experience to browse with it.